Hi, so in this video we're going to be comparing the three main types of RNA, which is mRNA, tRNA, and rRNA. So RNA is short for ribonucleic acid. Its structure is typically single-stranded. The three main types, messenger RNA, ribosomal RNA, and transfer RNA, are present in all organisms on Earth. However, there are different types of RNA present. These are just the three main ones. So typically, the nitrogenous bases in all three of these molecules are adenine, guanine, cytosine, and uracil. Of course, not thymine because that's only present in DNA. We will talk about later how tRNA may have additional altered bases as well. RNA plays vital roles in cellular processes like protein production, as well as it acts as the genetic code in certain viruses. An example of this is COVID-19, that is an RNA virus. So we're going to start off by looking at the central dogma, which in a very simplified version is just saying that DNA goes to RNA through transcription, and then RNA goes to a protein through translation. And there's many steps in between. So you can see the central dogma is on the right-hand side of this eukaryotic cell. This diagram is really good at depicting how transcription will occur in the nucleus, produces the mRNA. The mRNA will leave the nucleus and go to the ribosomes, which is now outside of the nucleus, and that is where translation will occur in order to produce that active protein. Here you can see, since it is a eukaryotic cell, there is pre-mRNA, which goes through post-transcription in order to become mRNA, and that just involves intron splicing. In prokaryotes, there is no pre-mRNA because they don't have introns to splice, but that's not important for the purpose of this video. So the first type that we're going to be talking about is messenger RNA, or mRNA. mRNA acts as the carrier pigeon of instructions. You can think about it as DNA is the original instruction manual. The mRNA is what carries these instructions outside of the nucleus into the ribosomes. It typically has a linear structure, however there are certain secondary structures as well. Again, in protein synthesis, mRNA is the product of transcription, and it is made in the nucleus, in eukaryotes that is. It contains codons from the DNA in the nucleus, and it carries it to the ribosomes, which are the site of protein translation. In translation, the mRNA will code a specific amino acid. It'll encode a specific amino acid sequence of a specific polypeptide, which just means that certain mRNAs are going to code for specific types of proteins. So in this diagram, that black arrow that just popped up is pointing to part of the ribosome, so that purplish pink thing, that is the ribosome, and the mRNA is getting threaded through, and that is where translation is occurring. The second type of RNA that we'll be talking about is ribosomal RNA. You can think of this as the ribosome producer or the factory machine builder, and this is because those ribosomes that the rRNA helps form along with protein is what's actually going to be the machine to translate those mRNA molecules into proteins. rRNA has a very complex structure. It has many loops and folds. It's sphere-shaped, and it's very interesting is that 80% of the total RNA present in a cell at any given time is rRNA. Like we already said, rRNA and protein is what ribosomes are made out of, and they do not possess anticodons or codons like the other two types of RNA that we're talking about do. So ribosomes are composed of a large subunit and a small subunit. and these make sure that the mRNA gets wedged in between during translation. Ribosomes are absolutely critical in order to synthesize proteins, and the structure and function of these ribosomes is very similar across all species. However, typically humans have more nucleotides in their rRNA than bacteria do. The last type of the most common RNA is transfer RNA, or tRNA. This is the delivery of the material. So you can think of this as like the UPS person dropping off your packages. tRNA has a cloverleaf structure, which is stabilized through strong hydrogen bonds between the nucleotides. And this is the example, if you remember earlier, we were talking about how they may have more bases than just the typical 4 in RNA. This is because those bases can be methylated to form like methylguanine or methylcytosine. And then this can make like these weird bases happen. tRNA is the smallest of the 3 main types of RNA. So if you remember, mRNA has codons, tRNA has anticodons, which works closely with the ribosome in order to read the codons and match up with them on the mRNA. So the main function of tRNA is to bring the amino acids, specific amino acids, remember, to the ribosomes so that they can be linked up to form proteins during the translation process. Each of the 20 amino acids has a specific tRNA. So it's going to match up exactly, and each of the tRNAs is going to contribute a different amino acid to the sequence. So it's not just one of the tRNA molecules being involved in translation. They're going to keep constantly switching out. So this diagram is just showing the 3 main types, I should say, of RNA in action. So blue circle here, messenger RNA. That will get fed through the ribosome, where the tRNA is going to, the anticodons on the tRNA is going to read the codons on the mRNA, and that's going to in turn keep adding those amino acids to the growing polypeptide chain. And these red circles here, that's showing that the rRNA is being formed, which will in turn build those ribosomes. So like I mentioned before, there are many other types of rRNA. Some of them are listed here. Like ribozymes are biological catalysts that are rRNA enzymes. There's double-stranded RNA, there's regulatory RNAs, there's snRNAs, there's many different types. They all have different functions. But in summary, those are the 3 main types. Just as a review, you can see this full diagram here, including the tRNA, the mRNA, and those ribosomes, which are made up of the rRNA plus protein. In summary again, mRNA carries those genetic codes from the DNA to the ribosomes. The ribosomal RNA helps build those ribosomes, which will work with the tRNA in order to bring specific amino acids to those ribosomes and build that polypeptide chain, which will eventually, once it's finished, form that protein. RNA is all about protein synthesis. So these 3 main types are involved very closely with protein synthesis, and cellular function would be impossible without it. Okay, and that concludes this video. Thank you.